Hello, and welcome back to the second half of acellular infectious agents. So we've been talking about viruses, and it seems only fitting that we talk about the novel coronaviruses. So coronaviruses, they all come from the same family. They are zoonoses, meaning they have zoonotic origins, meaning they are viruses of animals that have been transmitted to humans. There have been three pandemics in recent history. So SARS-CoV-1 was a 2003 pandemic that caused SARS, the disease, so severe acute respiratory syndrome. There was an initial outbreak in southern China, and so from an outbreak, it went to an epidemic, and then it spread to 27 countries, and it was ruled a pandemic. There were over 8,000 cases and 800 deaths. This gives this particular virus a case fatality rate of 10%. The reservoir, or original host, if you will, is a bat. Then the intermediate host was a civet, and then it was transferred from civets to humans, and then there was human-to-human transmission. The second coronavirus pandemic was MERS. MERS stands for Middle East Respiratory Syndrome. That pandemic occurred 2012 to 2016. First, there was an outbreak in Saudi Arabia, and then it spread to 25 countries and was declared a pandemic. There were over 1,800 cases and 600 deaths. This doesn't look like a very high number of cases, but if you see the deaths, this particular virus had a case fatality rate of 33%. So that means 1/3 of the people, 1/3 of the confirmed cases, they died. So this was a particular virus that was very virulent. It had a high mortality rate, so much higher than the SARS-CoV-1. Thankfully, it was not as infectious. The original host was once again a bat. The intermediate host was a camel. It transferred from camels to humans, and then human-to-human transmission. The current pandemic that we are in while I'm recording this is SARS-CoV-2. That's the name of the virus. So severe acute respiratory syndrome, coronavirus 2. The disease it causes is COVID-19. That is because the initial outbreak in Wuhan City, China was at the end of 2019. So there was an outbreak, it spread to an epidemic in China, and then it's currently spread to 202 countries. It was declared a pandemic in March of 2020. You can click here if you want to find the live counts from Johns Hopkins. Looking at it today, April 14th, there have been 1,970,879 confirmed cases of COVID-19. Of those, 125,678 have died. So that gives this particular pandemic, this virus, a case fatality rate of 6.3 if you do the math. The true mortality rate, or case fatality rate, is probably going to turn out to be lower, and that has to do with initial testing. There were limited tests, so only the sickest of people were tested in several countries, and so this kind of makes it look like this particular virus is more virulent than it is. It will probably shake out to be in single digits, 1 to 2%. The reservoir is once again a bat. This virus shares 97% sequence homology to a current bat coronavirus. They do not know if there's an intermediate host at this point. There's lots of conjectures. They thought it was a pangolin. They have since ruled that out. There's studies being done in labs, and so they know certain species that it doesn't grow well, and it doesn't grow well in dogs, pigs, chickens, and ducks, so they've ruled those out. They do know that it can multiply pretty efficiently in cats and ferrets. That doesn't mean that either one of them was the intermediate host or that cats are spreading coronavirus, so don't go kill your cat. All they know is that from bats, there may have been an intermediate host, there may have not have been, but now we have transmission, community cases, so from person to person. So if we look at the structure of SARS-CoV-2, it is 78 nanometers. So remember, we measure viruses in nanometers, not micrometers. They can only be seen by electron microscope. So it falls within a range of about 20 to 150 nanometers, so like most viruses. It has a fairly large genome, 26 to 32 kilobases, so it's pretty large for an RNA virus. It is single-stranded RNA. There are 5 structural proteins and 60 non-structural proteins coded for in that genome. So if we look at this picture, we see those spike proteins we were talking about that are there for attachment. This is also what they're using for vaccine development. There's an M protein and there's E proteins associated with the envelope. And then also there's this HE protein, which they believe assists and is responsible for viral entry. There is a helical capsid and a single piece of RNA, so it is not in segments like the flu, so there won't be any sort of antigenic shift. So all of these changes in these coronaviruses are going to be due to drift or just slow mutations. So if there's changes in the spike protein, that would be antigenic drift, which there has been, which we'll talk a little bit more about in just a second. So it's an RNA virus. And so there you see its genetic material. So let's just remind you of the steps in viral infection. So there is transmission, we know by respiratory droplets. They believe by fomites that to just recent data, maybe not as much as we thought. And then they were believing too that possibly through the fecal oral route. But the number one way it's going to be spread is by respiratory droplets. Then there is attachment to those angiotensin II receptors on cells of the mucosa of the respiratory tract. There's also connection to or attachment to cells lining the gastrointestinal tract, so that is new from the original SARS. That did not occur with SARS-CoV-1. Also, there is binding to those receptors in blood vessels and also in the kidney. Entry has to occur, and that's by penetration through endocytosis. And then, of course, there's uncoding. So the single-stranded RNA then can be used as messenger RNA in the cytoplasm. And then also it can be copied by that RNA-dependent RNA polymerase carried by the virus. And that's the sloppy one. Assembly is going to occur spontaneously in the cytoplasm, and this particular virus releases by budding. When viruses release, they can infect their neighboring cells, or they can be transferred through the bloodstream or through the lymphatic system. The majority of the damage is going to be done through collateral damage and through our immune response. And so it's really just going to be the droplets that seem to be the number one spreader as of current data. This, of course, was preliminary data, and you've probably heard all about that in the news. Just do continue with hand washing. If you're watching this recently, continue with your hand washing. So stages of infection, we've looked at this slide before as well. There's an incubation period, and so that's going to be the time between exposure to the onset of disease. And they're thinking it could be anywhere from 2 to possibly 10 days. 14 days is what they're setting it at for self-quarantine. And then the next stage would be illness. And we know that with a lot of diseases, this one including, there's a prodromal phase. And so this is when somebody has been exposed. They're in the incubation period. They don't quite have all the signs and symptoms yet, but they just sort of feel a little off, little run down. And then once you have illness, that's when you're actually, the signs, the things that you can see. Remember, those are things you can measure, like a fever. Ace of base, I can't remember the year, somewhere in the 80s. Anyway, so signs are things that you can see. So with COVID-19, things that you can see, you're going to see a fever, you're going to see a dry cough. Symptoms would be other things like fatigue, just that overall feeling, oh, I don't know, maybe like this. So malaise, body aches. Those are things that would fall under symptoms, but those are the common ones for COVID-19. As the disease progresses, eventually about 7 to 10 days, someone's going to feel better, and then that's called convalescence. So that's recuperation and recovery from the disease. So the big thing is, when are people infectious? So it seems that for COVID-19, the period of highest infectivity for symptomatic cases, of course, ranges from 2 days before the onset of symptoms to up to 3 days after their resolution. The exact limits are still under investigation, but what the recent things that they found out is that 10 days after symptoms, although someone will still have viral RNA in their sputum, they think that there's little risk of infectivity. 2 weeks after exposure, the adaptive immune system will start to produce antibodies. So that's called seroconversion. So that's what they're talking about with that antibody test. And I'll bring that up again when we discuss the immune system. Okay, so 70% of the people is asymptomatic, 30% of the people have symptoms, and about those 10% is the latest I've heard are going to need hospital care. And so that's really why we're in lockdown, because that 10%, that's a lot of people. So those people will need ventilators, and they can end up in the ICU. And so what's happening, if we kind of look over here, we talked about the receptor already, angioconverting enzyme 2. The target group of people that are having the hardest time with this disease and the highest mortality, these are going to be the elderly, so people over 65 years of age, or people with underlying disease, so people that already have hypertension, they already have COPD, they're immunocompromised, they have diabetes, they have cardiovascular disease. The problems with this particular disease is that it does lead to hypoxia, and that's really what gets people to go to the hospital. If you have trouble breathing, your lips turn blue, that's going to get you to go to the hospital pretty quickly. What can happen, though, too, what they find out about 9 to 10 days later, which is causing the problem, is the body's reaction. So there's this cytokine storm, which leads to septic shock. And so you end up with metabolic acidosis, coagulation dysfunction, and we talked about this, that disseminated intravascular clotting is throwing blood clots to the lungs, to the liver, to the kidneys, and you get multiple organ failure. So as opposed to the first SARS, where people were dying more from the respiratory distress, acute respiratory distress syndrome, this particular virus, it seems to be more of these septic complications due to that cytokine storm. The positive news is that they're getting better at treating it. They're realizing that a lot of this is being brought about by interleukin 6, which is a chemical messenger of the immune system. And so some pretty good therapy for these particularly critical patients is giving an IL-2 blocker. So in the US, they are getting much better at treating it, so hoping to see that mortality rate go down. And so I'm just kind of showing you this. It has cytokine storm, may cause tissue damage. This was for the original SARS and MERS. But you see that you don't really see that it was more the respiratory distress or pneumonia that was causing death in these patients. I just kind of showed you, this is just showing you an air sac in the lung, and these type 2 surfactant secreting cells have high receptors for that SARS-CoV-2 virus. Okay, and hopefully you kind of remember this from earlier in the semester. We talked about it in regards to bacteremia. Viremia can do the same thing. You get viremia, sepsis, so that over-exaggerated inflammatory response that leads to septic shock, drop in blood pressure, clotting, organ damage, and death. Okay, so categories of animal viral infections by time course. So just any acute infection, they develop quickly and they're over quickly, and this is going to be due to burst of virions in this case from the infected host cells. The immune system gradually eliminates this virus, and so that's what marks it as an acute infection. It comes on quickly, it's over quickly, and the virus goes away. So that's the unique thing about an acute viral infection. And our poster child for that one is influenza. The other types, you have acute infections and you have persistent infections. And so these persistent infections will persist. They can persist for years or a lifetime with or without symptoms. So there's chronic infections and latent infections. So we'll look at chronic infections first. So these are when there is a continuous production of low levels of virus particles. So this could be for decades or a lifetime. The carriers in this state, they can lack symptoms, but they're still transmitting the virus. And so there's virus shedding, but it's not all at once. So there's not a whole lot of cells dying at one time. Like I said, usually this is going to be asymptomatic. And so the poster child for this one is hepatitis B. So it's showing you incubation period, disease, and then there are no symptoms, but there's just this constant, steady release of virus. So these people are carriers. And so I gave you this table. So hepatitis B, that's going to be our poster child for a chronic viral infection. Hepatitis, that word, hepato means liver, itis means inflammation of. So hepatitis B. Hepatitis C is also a chronic infection. Hepatitis B we have a vaccine for. It is high in Asia and in Africa. Hepatitis C is more prevalent in the US. We have drugs now for hepatitis C, and they're pretty effective. And so a person can now be cured of hepatitis C and not have that chronic infection for life. And so they consider someone to be cured of hepatitis C when virus number has not been detected in blood for about 3 months. But both of those cause inflammation of the liver, get cirrhosis, which is that end-stage death of a liver if it's left untreated. It also is going to lead to cancer, so liver cancer. We're going to talk about that one a little bit later. So hepatitis B, that's our example of a chronic viral infection. So latent viral infections, like other infections we've talked about, that means you have to wait. It means there's initial infection, it goes away, and then it comes back. So this is when a virus is going to form a provirus, meaning a silent viral genome. So the viral genome is going to be inserted into the host cell chromosome, and it's going to be replicated either with that chromosome, or it can even be sort of separately like a plasmid. The big difference between an acute and a latent infection is that this cannot be eliminated from the body, so it's not eliminated. The thing is, it can go quiet and dormant, so latent means to wait, but it can later be reactivated. And so 2 examples for that. One is going to be herpes simplex 1. So you're infected as a child, you get a cold sore, and then that virus is going to form a provirus, and it's going to hide. It's going to hide in sensory cranial nerves. And then later in life, and not in everybody, only in some people, perhaps when someone actually has a cold, so their immune system is challenged, or when they get older or stressed, then that virus can reactivate, and then someone can get cold sores. We used varicella zoster virus, the virus that causes chickenpox, as an example of a latent infection. We talked about it earlier this semester. That when a person ages, their immune system goes down, it can become reactivated, and a person can get shingles. So those, notice these are herpes and the varicella zoster virus. They are all in the same family, so herpes viridae family. And like I said, those are all examples of latent viruses. So herpes simplex 2, that's genital herpes, that works the same way as the cold sores. Okay, so our last one, HIV, is the virus that causes AIDS, so human immunodeficiency virus. And your book kind of billed it as a mixed virus, and it has a unique lifestyle, and since so many people are infected with HIV, you're going to have patients infected with HIV, we should probably learn a little bit about its life cycle.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell, where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
Okay, so that is a unique life cycle. Attachment, once again, is going to be very specific. So the CD4 marker first on macrophages, where CCR5 is that co-receptor, and then later on in the infection, it's going to be CD4 and CRCX4 is the T helper cells. So it's the attachment that's always going to determine host range and tropism. Then, of course, we have attachment, entry, then synthesis. This is a retrovirus, meaning it's going to go backwards. It carries its own reverse transcriptase. So it's going to go from RNA to DNA. It carries a viral protein integrase, which is going to then allow that DNA to integrate into host DNA and form a provirus. So you cannot get rid of it. There is an acute initial infection with high virus particles. There's signs and symptoms. They're kind of flu-like, swollen glands, nothing that would tip you off that you've been infected with HIV. But there is an acute initial infection. And then there's this chronic shedding of virus from macrophages, dendritic cells. I don't know from AP1 if you remember those microglial cells in the brain. But they're basically going to serve as just this constant chronic shed of the virus. There's normally no weakening really of the immune system that's really apparent at that stage. You are losing macrophages and dendritic cells, but it's not until the virus infects the T helper cells. And so we're going to talk about that when we talk about the immune system. When those T helper cells start to die, a person loses their adaptive immune system. And so when those numbers go to 200 cells or below per microliter, and just telling you normal is about 600 to 1200 cells per microliter, that's when someone has acquired immunodeficiency syndrome. So their immune system is no longer functioning, and that's when they're going to succumb to a myriad of opportunistic infections. We talk about fungal infections and bacterial infections that really hit AIDS patients. Half of the untreated patients are going to progress to AIDS within 9 to 10 years. Pretend there's an S there. Some people, though, have very low viral set points, meaning they are not shedding high amounts of virus. They can actually survive for many years, even without treatment. So viruses are associated with cancer. Cancer is unregulated cell growth. So that's really what's occurring when someone has a tumor. There's two types of tumors, malignant and benign. A malignant tumor is one that has its own blood supply, or it has occurred so many mutations that it has lost its cellular attachments and is now spread to another part of the body. Proto means original, oncogenes. These are normal genes that are involved in stimulating cell growth. So think of them, I'm giving you this car analogy. So think of them like go signals, so the gas that cause cells to go through mitosis. If there's mutations in these proto-oncogenes that can cause them to basically stimulate growth, so unregulated cell growth, so too much cell growth, then it's called an oncogene. So an oncogene can cause the growth of cancer cells. In the body, there are multiple tumor suppressor genes. So these are genes, a lot of them are going to be proofreading proteins. A big one is going to be P53. And so these are genes that are going to inhibit growth and division when there are mutations. So I like this little picture. It's just kind of showing you normal genes that regulate cell growth when they're working, then cancer does not occur. The first mutation can lead to accelerated cell division. Okay, so notice we're not at cancer yet. If there are tumor suppressors that are functioning, they'll take care of that first mutation, and it will just go back to normal cellular growth. If there is an absence of tumor suppressors, so functional tumor suppressors, think about it like a car that doesn't have any brakes. So you have an active oncogene stimulating cell growth, and now you don't have any brakes. And so that's how one ends up with a tumor and cancer. So it's not just one mutation, it takes multiple mutations, and you have to go around the regular regulation of cell growth in order to get to cancer. So mutations in these genes, go or stop, cause abnormal or uncontrolled growth. And so usually, like I said, multiple changes at different sites, either in the proto-oncogenes or in tumor suppressors. So that's usually why cancer occurs when someone's older. How viruses are causing this conversion between proto-oncogenes to oncogenes is an insertion of a provirus. It can disrupt a gene that would slow things down. It could basically bind by a promoter and really stimulate production of a particular oncogene, or it just could be cause of damage to the DNA, and then there's inflammation, and then you get unregulated cell growth. Viruses too, sometimes they carry an oncogene themselves, which stimulate a cell to grow. Human papillomaviruses are associated with cervical cancer most of all. So all cases of cervical cancer almost are associated with a prior human papillomavirus infection. That's because human papillomavirus, it takes out the tumor suppressor genes, and so you lose that proofreading ability. So it really messes with p53 apoptosis, so you end up with unregulated cell growth. It's also involved in other types of cancers as well. But they've determined about 13% of all cancers are caused by oncogenic viruses. So looking at this chart that I've given you, you see that all of these viruses are associated with persistent infection. So there's going to be provirus formation. So these are viruses that the body cannot clear. The majority of them are going to be DNA viruses. And so we're going to watch YouTube about Epstein-Barr virus. We talked about hepatitis B. Hepatitis B virus is that chronic infection. It is linked to liver cancer. Herpes virus, which you can never get rid of, it's the gift that keeps on giving. It can cause Kaposi sarcoma. This is one that for most people, it is not a problem. This is a different type of herpes, so type 8, so not cold sores or genital herpes, but this is going to be common in AIDS patients. They've lost their immune system, so they're going to get this type of cancer. Human papillomavirus, we talked about that. It's involved in almost all cervical cancers. There's now a vaccine to this virus, so the Gardasil vaccine, so hopefully we will eradicate cervical cancer. Hepatitis C is also involved in liver cancer. And then there's also HTVL1, which is human T-cell lymphotropic virus, which causes adult T-cell leukemia. And those are rare. So just kind of showing you all viruses associated with cancers are persistent infections. If you have this productive infection, notice there is no tumor formation here. Over here, it has to be a viral DNA that integrates into the host DNA, either in the chromosome itself, or it can replicate separately as a plasmid, but only through that latent infection can you get tumor growth, than it being a oncogenic virus.
When we talked about the way that EBV infection works, we actually simplified things a little. So let's go back and be a little more precise now. Anytime EBV infects a new cell, whether it's epithelial cells, B cells, or T cells, it can go one of two ways, lytic or latent. Now with B cells, it's almost always latent, but either way, when the infection is latent, the viral genome sits in the nucleus and a small subset of viral genes are transcribed. And remember that, the fact that it's latent doesn't mean no viral genes are transcribed, it just means no full viral particles are produced. So these genes that are transcribed, what do? Well, it turns out they tell the cell to stay alive and proliferate, instead of dying after normal lifespan, which is usual. And the way they do this is to down-regulate apoptosis and upregulate various proliferative pathways. Why would the virus do that? Because if the B cell survives, then the virus inside it survives. And if the B cell replicates, then it replicates the viral DNA in its nucleus too. So this is actually quite smart of the virus. Now there is a downside to producing these viral proteins during latency. which is that cytotoxic T cells can still recognize and kill latently infected B cells. But nevertheless, by the end of an acute infection with EBV, there will be a small population of latently infected B cells that escapes the immune system and survives long term as the immune response dials down. Okay, so increased survival and increased proliferation. Now, what does that make you think of? Hopefully cancer. And indeed, because of these viral proteins, EBV infection predisposes to cancer. And you might already have known that because it's a gamma herpes virus, and these can induce cancer. Now, what kinds of cancer does EBV cause? Well, just remember the cells that EBV infects. Epithelial cells, so that can lead to nasopharyngeal carcinoma. B cells, which can lead to Hodgkin lymphoma and non-Hodgkin B-cell lymphomas, especially Burkitt's lymphoma. And T cells, which can lead to T-cell lymphomas. Now, you might not know what all these different cancers are exactly, and that's fine, but at least understand the underlying mechanism. Now, something that's interesting is that people who are immunosuppressed, especially people with HIV, get a lot more of these cancers, especially the lymphomas. And now that we've talked about all the mechanisms, the reason should be clear. The more immunosuppressed you are, the more EBV can disseminate, the more it'll produce its genes that contribute to the formation of cancers.
Okay, so that was just a sort of a nice, I like the way he said that he saw that it downregulates apoptosis and upregulates cell division, and so that really, in a nutshell, that is the definition of what tumors do. The initial infection, many people will never even notice that they've been infected by the Epstein-Barr virus. If it happens in children, it's either asymptomatic or, nothing that you would attribute to anything, so just minor symptoms that go. If someone is infected as an adolescent, they have about a 35 to 50% chance of developing mononucleosis. So mono, the kissing disease, not sure that you've heard about it, but it just causes usual fever, body aches, incredible fatigue. Like I said, it starts sort of as an upper respiratory thing. But this is one, like I said, example of a latent infection that can then become reactivated. And then basically cause cancer. So it's just sort of constantly. So this is an example of a persistent infection that is never cleared from the body. And so this virus is being reproduced every time the cell is produced, and there's just low levels of viral proteins being made that are basically going around the controls for cell growth to get unregulated cell growth, and it leads to lymphomas. Okay, so just a couple more topics. So cultivating viruses. So since viruses, they require a host, you cannot grow them by themselves, so they need a host. So there's special methods that have to be used. The reason someone would want to grow viruses, of course, to isolate, identify viruses in clinical specimens, to prepare viruses for vaccine development, and also to do research. So methods used to grow viruses, they were grown in animals, so mice and rats. When I was working on an influenza virus project, we grew our virus in embryonated chicken eggs. So these are eggs that have been fertilized, and you incubate them, and then you just draw out the virus in the fluid. Another method that's a little neater and cleaner is going to be through cell culture or tissue culture. And so there are flat flasks, tissue cells. They're going to grow in sheets called monolayers, and then you can just add your virus to the media, and then assuming there's a match between virus and host cell, then you're going to get a viral infection. And so that's really kind of what's commonly used. Since virus can be grown in culture, so in vitro, one can visualize the cytopathic effects of viruses. And so what happens to cells when they become infected with virus, they can change shape, they can detach from the surface, so they're no longer growing in sheets attached to the culture flask, they're not in a monolayer anymore. They can lyse, so they can die. They can also, the next two I'm going to show you in this next slide. Another thing that can happen is they can fuse and form a syncytium. So this is actually a histological slide from a pap smear showing a herpes simplex virus infection. And so this is what a normal cell looks like, and this is one that's sort of in transition. You can see that that nucleus does not look the same. It's starting to stain more darkly. But this right here is a megacell, basically a cellular syncytium. So you have multiple nuclei. You also see these sort of darkly staining patches in these cells as well, and these are called inclusion bodies, and that could be sites of viral replication. So whenever there's genetic material, it always absorbs stain really darkly. You might observe the same sort of thing in cells that look similar with the varicella zoster virus, that respiratory syncytial virus, so RSV, will also form these sort of cells, and that's how that virus got its name. So a lot of what we know about viruses was discovered using phages, so viruses that infect bacteria. And so there's two basic types of life cycles. There's a lytic cycle, which brings about a productive infection, which means new virus particles are going to be released. And there's a lysogenic cycle, which is a latent infection. And so just like we were talking about latent infections in animal cells, it just means wait. So we'll look at the lytic cycle first. And so the phages that can only go through a lytic cycle are called virulent phages. And so we're going to look at the life cycle for T4. A temperate phage, so the poster child for that is lambda, is one that can go through the lysogenic cycle, or it can switch and go through the lytic cycle. So they're capable of both lysogeny and also going through the lytic cycle. So instead of a provirus, like we talked about with animal viruses, temperate phages in a lysogenic cycle will form a prophage. And so once again, the DNA is going to be inserted into the host cell genome and replicated every time the host cell divides. A bacterial cell that has integrated phage DNA is called a lysogen. What can occur if it's a temperate phage is that you can get what's called phage induction, meaning this phage DNA can leave the chromosome and then go through that lytic cycle. So then it can lyse cells and have a productive infection. So that's something that's unique to phages. And so we looked at these pictures already. So you still have attachment, genome entry, synthesis, assembly, and release. And so when you have the release, that's why it's called a productive infection. So T4 is the model for that. The higher process takes about 30 minutes long. The T4 is going to release about 200 virions every time it lyses a cell. So you can get a whole lot of virus really, really quickly.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lyses, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
Okay, so this is a diagram just showing you everything that YouTube just showed you. Lambda can go through that lytic cycle, so attachment, entry, synthesis, assembly, and then release. Or it can go through that lysogenic cycle where it becomes a prophage, then it's going to be copied each time that cell divides, and then there can be that phage induction, and this can be brought about by stress to the cell. If I wanted to get my virus out from lambda in the lab, I would hit it with UV light. So a prophage can begin the process of lytic infection based on that metabolic state of the host cell, so that can influence that. And then once we have excision, then it goes through synthesis, assembly, and then release. When a cell is a lysogen, meaning when it is carrying prophage DNA, it is immune to reinfection. There's going to be repressors that are going to keep that phage integrated, but then that cell is also just going to be protected from further viral infection, so it can't be super infected. We talked about this when we talked about bacteria. So some bacteria have their virulence carry toxin genes that were originally brought to them by a phage, so by a bacteriophage. And so these are when a bacteria has a prophage and it gives it a new characteristic or trait, then it's called lysogenic conversion. So Clostridium botulinum, that bacteria, the only ones that are virulent are ones that are have that integrated prophage carrying the botulinum toxin. And so we looked at with botulism, diphtheria, the Shiga toxin in that O157H7 E. coli, Salmonella enterica that causes food poisoning, just got a modification of their LPS. And then Streptococcus pyogenes gets that pyrogenic exotoxin from a phage virus, and the toxin that causes cholera and Vibrio cholerae also is from a phage. So just whenever that prophage is giving a bacteria a new trait, then it's called lysogenic conversion. So it's just a nice summation. Some of our virions are that extracellular state of a virus. You get infection of the host cell. You're either going to have a productive infection or it's going to go to the latent state. Productive infections, you can get lysis, which is going to kill the host cell, or you can get slow release where the host cells do not lyse and that host cell is going to continuously release the virus. In that latent state, we are going to get the viral DNA is going to either integrate or just replicate aside from the chromosome as a plasmid. Host cell multiplies, but phenotype often changed because of viral genes, and that was called that lysogenic conversion. So it could be virulence factors. We talked about how antibiotic resistance can also be transferred to a bacteria that way, or it can just be another gene. Just tying it back to our study of bacteria. Okay, so just like there were methods to quantitate bacteria in a sample, there are also methods for quantitating viruses or phages. We're just looking at a method for quantitating phages. So this would be called a plaque assay. And so plaque assays are used to count phage particles in samples such as sewage, seawater, or soil. And so if you take bacteria and you grow them on an agar plate, then they'll form what's called a lawn. So this is solid bacteria. If you take agar and you take it when it's warm but not hot, so it's still liquid, and you mix the bacteria with phage particles, and then you pour that agar onto a plate and incubate it, what you're going to find is you'll find these sort of empty places called plaques. So plaques represent what's called a plaque forming unit. And so that's sort of equal to saying that one virus particle infected the bacteria here, they cause lysis, and they spread to the adjacent bacteria. And so that's how phages are quantitated. So you would count the plaques to yield the titer, and then you could then calculate the concentration of phage in an original sample. So our last topic is prions. So that stands for proteinaceous infectious particles. And we talked a little bit about this way at the beginning of the semester. But these are composed completely of proteins, so they are not made of nucleic acids. What they cause are slow, fatal, incurable human diseases of the central nervous system. They're called spongiform encephalopathies because when you look at a prion-infected brain, there are these lesions that basically they look like holes, so that's why they got the name spongiform encephalopathies. They're usually transmitted only within a species, but however, mad cow's disease, also called bovine spongiform encephalitis, it killed more than 170 people in England, and they also believe that cats have a similar disease that has been transferred from cat food that possibly had infected cow brain in it. So I called prions before good proteins gone bad, or I could say good proteins folded badly. And so they did a lot of the work in a disease that affects sheep and goat called scrapie. And so these names, so this is prion, protein, cellular. Prions, there's a normal functioning cell protein, so in neurons in the brain, and they're folded a particular way. And so what a prion is, it is a misfolded protein. So in this case, in this picture, PRPSC just means prion protein scrapie. It is same amino acid sequence, it is just folded differently. What's weird is that this protein can cause this protein to misfold. And to be honest, they don't know why that works. And then you get these insoluble aggregates that basically form these lesions inside a brain. Some people are afraid of spiders. I'm afraid of prions because prions, one thing, I would not want to die from one of these diseases, but they are resistant to proteases even though they're proteins, so you can't cut them up. They're resistant to heat and chemical treatments, so they're very hard to get rid of. And I guess what scares me about them, if you look at the animal diseases, there are animals that are not eating other animals or brain that are getting them. And so they don't really know a lot about these prions and exactly where they come from. What they've determined in the human population, yes, you can always get them by eating infected brain. And they learned that by studying the Thru tribe, because one of their burial rites is that you eat the brain of your ancestors. And it was mostly the women and children that got to eat the brain of their dead, departed ancestors, and they developed Kuru, so a prion disease. Creutzfeldt-Jakob disease, there is an inherited form of it, but then they came up with variant Creutzfeldt-Jakob disease because this, they determined, came from not from being inherited, but from eating the meat or beef that was contaminated with bovine spongiform encephalopathy prion, so from mad cow meat, basically. There's a couple other inherited ones, Gertzmann, Straussel, Schenker, that's just a great name, just good old German name there. And then fatal familial insomnia. So you get them two ways, they're inherited or from eating infected meat or brain, and it causes spongiform encephalopathies. And so that's kind of the big thing. As far as I know, it's not been transferred directly human to human. However, that's what occurs in animals, and so that's the part that keeps me up at night. This wraps it up. I hope you've enjoyed it. Have a great day, and see you around.